How is everyone doing?
Good.
We got one good and a bunch of people who have just died or something.
Sorry, what was the question?
How are you doing?
We're floating
Cool.
on a raging sea.
Yes, we are. So, I think it's another time for us to kind of step back and do a checkup on how you're feeling about the class, your skills with R, all that kind of stuff, kind of see what we can work on to get you up to speed if you're feeling left behind on anything. And I just had planned on doing some live coding today. I'll try to go pretty slow, and I can post all that that script that I eventually come up with afterwards. But I need a starting point. Feel free to just jump in. What's something you're struggling with?
So I'm in one of the assignments, I don't remember which one it was, but it said I got to use GLM and AOV, and I'm still confused on what's the difference between those two.
Okay.
Yeah.
All right. I'm making a note of these so I can get to them all. But somebody else, so I don't get sidetracked, I'm just going to make a list of stuff. So yell at me. What else? We got GLM versus AOV.
So I have a question that might not necessarily be related to the coding specifically, but I'm having a hard time finding what to do for my project.
Okay.
What's a good direction to go for finding like a project? I guess I mostly like, it feels like the project's very open-ended with like a lot of unlimited possibilities. And I have a hard time with unlimited possibilities. I'm a point and do that kind of analyzer.
You want me to give you something? I'm going to make it really hard.
No, I don't know if I want you to give me something. I don't know about that. Okay. Just like a little guidance.
Okay, what's your interest?
Oh man, that's also a very open-ended question.
It doesn't have to be biology either, right? I've had people do this on stock market stuff. I've had people do it on baseball. I've had them do it on genomic data.
Genomic data is kind of what I'm most interested in, but I feel like if you were to say, hey, do an R project on genomic data, I would feel like I was in a sea of unknown.
Right.
Scary.
Okay, so I guess the question kind of should be for everyone when you're picking your data, what data analysis skills do you really want to have in your back pocket, right? So would you like to learn how to make a phylogeny?
Yeah, that sounds super interesting.
Okay, so we can do that. So I will put that down, finding genomic data for phylogeny. I think probably you're not alone in that interest, so that'll probably benefit a lot of people. All right, what else?
All right. One concern I had is in modeling, what exactly ad predictions creates and why my predictions don't look like predictions.
They don't look like predictions?
No.
Okay. So what does ad predictions actually do? Okay. Just so I kind of am on the right page, what do your predictions look like?
If I try plotting my predictions, like they'll end up in a totally different cluster than the actual data.
Yeah, okay. All right. And that can happen. That means your model was bad. That's all.
Okay.
Yeah. So we'll take another look at what ad predictions is actually doing and how those plots relate to reality of our data. Okay.
Sounds good.
I have one. It was not too long ago. We were looking at the ANOVA tables and how to read those.
Mm.
And there was one ANOVA table that didn't pop up with a p-value.
Yeah.
I don't know how to read that really bad.
Okay, if it doesn't pop up with a p-value, typically that means that it was basically unable to fit things correctly, or where the authors of that function decided to let you do it anyway and just show you that nothing worked. But we can look at that. How to read analysis of variance table. And with that, I'm actually going to show you another post hoc trick. So a Tukey test. Okay. Who else?
When making models, I remember we used, or did we, the STEP AI? My question was like, I remember like we just essentially give it a model to start with, and then from that it'll like go off and make a bunch of variations. But like, is there a way of determining like which model to give it first or right off the bat, or we just do the whole like give them all plus signs or all multiplications? Like, you know, like, is there a starting point before we feed it that?
I'll give you the short answer right now, but we'll talk about it more. No, there's no. It's the shortest answer possible in English. So yeah. Okay. But yeah, we'll take a look at how to pick an initial model, what's reasonable and what isn't. Because if you give too much to your model, it does what's called overfitting. So I'll talk about that too. Come on, people, give me more. I know I'm not like the best teacher on earth and fully understand everything we've talked about. That's not possible.
Let's see. One thing might be handling date data.
Okay.
Formats of that. That's something that I've kind of come across a little bit.
Gotcha. There are several different classes of date data that R is capable of using. The best one is probably called POSIX CT. And so we'll look at that particular class.
All right.
And how to wrangle dates into it. What's nice about that one is it actually will take into account like time zones and leap years and all that kind of stuff. Speak now or forever hold your peace here. I can sit here awkwardly staring at these black boxes all day long. It's like a superpower.
Can we make a R code that would tell us who's going to win the election, or no?
Apparently not. Nobody can do that.
Or was their problem that they used Python and they didn't use R? Was that the whole?
They were using R and,
well, never mind.
Data. Yeah.
The most beautiful program.
It doesn't matter what you're using if your model isn't reflecting reality. Yep. All right. Well, I guess that's enough to get started with. So let me share my screen. Okay, you see that?
Yep.
Okay. I'm gonna open a new project, or an old project, new session. Where is it? Oh yeah, on my desktop. Make this a bit bigger. Okay. So first off, we've got GLM versus analysis of variance. So let's start a new script here. All right, so we've got tidyverse and the model R packages. Let's see. What data set? Let's go with the biolog plate data. Okay, so we'll read that in. Take a quick look at it just so we remember what's going on with it. Okay, so we've got sample ID, replicate, well, dilution, substrate, and then hour 24, 48, and 144. What's the first thing we got to do with this data set? Yeah. Okay. So df pivot longer. Okay, we want the calls and then names to, values to, and then the names prefix. The columns are the last three. We've got 8 in there, but to be safe, I'm going to say starts with HR underscore. Oops, I spelled starts with 2 S's. Okay. Okay, so those are the columns that we're going to be stacking on top of each other. The names to, what do we want to call that? Time? Sure. And the values to absorbance, if you remember this old data set. Let's see if that did it for us. Yep, there it is. Okay, so that's in nice long format. Time should be a number though, because we want to plot it continuously. So let's get rid of HR on there by adding one more little line here. Names prefix HR underscore. All right, that should do it. Okay, so we've got sample ID is a character, replicate is a double. That should actually be a factor because you can't have 1.5 replicates. Well is character, sure. Dilution is a double, yep. Substrate character, time character, and absorbance double. So let's change replicate into a factor and change time into a numeric. So I'll just go ahead and add another pipe here and say mutate rep equals as factor rep. Okay. And time equals as numeric time. Okay, that's a factor now. That's a double. That's great. So I want to save this. I'm just going to overwrite everything. I'm just going to say df is all of that. Okay, so now if I look at df, it's ready to go. So first thing, let's do a little quick plot so we kind of remember what we're talking about with this data set. We're going to plot df, the aesthetic, x should be time, y should be absorbance. Let's do color is dilution plus geom smooth plus facet rep as a function of substrate. So this will be a big looking plot. We'll see what we get here. Let's change color dilution to factor dilution. See if that fixes it. We should have 3 colors because there were 3 dilution values. There we go. And I'll get rid of the standard error by saying SE equals false. Those gray lines are just confusing. Okay, so here's our plot. Over time for the 3 dilutions, for each of those carbon substrates, how they were utilized. Of course, this is all of the samples together. So sample ID has just been all mixed together. So we've got 4 different sources, 3 replicates for each. So this isn't looking at sample ID, but there we have it. What hypothesis might come to mind for you if you're looking at this? What should we test with our model?
Just absorbance as a function of time and substrate, or yeah.
Okay. So what's your hypothesis in that case?
All right. With increasing time, increased absorbance. And then as far as the substrates go,
well, we got a few choices, but the easiest one and the one that is most often kind of tested is, are they different? Right? Where our null hypothesis would be there is no difference between substrates. Okay, so we'll run a test and we'll either reject that null or fail to reject it.
All right.
So we can test that. All right. So let's see here. Let's start out with a GLM, a generalized linear model. And here's our formula. Well, first I'm going to give it the data equals df. Formula equals absorbance as a function of, and we've got substrate and time. Okay, so that's mod 1. All right. To get a look inside there, we do summary. Okay, that's a bunch of stuff. So what's going on here?
Is it showing you what substrates are significantly different?
Yep. And then way down here at the bottom, it's alphabetical in this case, is time. And that's significant. Okay. Our GLM, it's not able to model substrate as if it were continuous, right? Because it's not. These are categories. And so basically what it's doing behind the scenes is it's looking at that slope for time and overall, and it said, okay, yep, time's important. Then what it did was subset your data to each substrate. Okay, so it did a subset down to D-mannitol, and it looked at time for just that one. All right, so let's see. Here it is. Okay, and so we found that D-mannitol was significantly different from the overall slope of time for everything. Okay, so the average increase over time versus D-mannitol or malic acid. Okay, you can see here malic acid, it does go up over time, but it's not significantly different from the average effect of time overall. Okay. If I do this same thing here, but instead of GLM, I put AOV. Oops, I called that mod 1 again. Meant mod 2. Here's our analysis of variance table. What's different about this?
It kind of reduces substrate all down to like one slope, I guess, or?
Yeah, pretty much. It's basically saying, are the substrates different from each other? Yes. Okay, so we reject that null hypothesis. Are the times different from each other? Yes, we reject that. So it's telling us substrate and time were important. Now, if I throw in a multiplier here, it's also going to give us this interaction. Do the substrates behave differently with respect to time? Well, in this case, yes. And that makes sense. Look at water. Nothing versus tween 80. Goes a nice straight line up. Okay. So the general linear model, if you give it one predictor, will give you the same test as an analysis of variance. Okay. So ANOVA, this AOV, is actually just a special case of a generalized linear model. And so I often don't use it. I usually just go straight to GLM. But sometimes in a quick look, it can just be nice to know whether that particular factor was significant. Whereas here, it's going to break it down for you and say which ones were different from the mean. So let's look GLM as a function of substrate by itself, and then AOV as a function of substrate by itself. AOV is just saying, yes, substrate is significant as a predictor, and the generalized linear model is saying, yes, but only some of them. Okay. Does that help answer the question about the difference between GLM and AOV?
Yeah, it does.
Okay, it gets to be a pretty kind of academic, math-heavy description. I don't want to go in too much, but I think the take-home can be that analysis of variance is actually just a special case, a very specific version of a GLM. In fact, all the statistical tests you learn about, like a t-test, chi-squared, all that, they're all regression models in the back. They're just special cases of regression. In most cases that you'll encounter, you'll be able to kind of substitute a t-test or whatever with a general linear model and get the same results, but a little bit more detail. Okay, I'm going to save the finding genomic data for later because we already got this pulled up. So let's take a look at the add predictions. Okay, so I'm going to dump this model. First, let's look at ANOVA. Are these models different? Mod 1, mod 2. It can't tell them apart. Okay. No big surprise. They're the same thing. All right. So I'm going to dump that one. And I'm going to change this model and make it a bit more ridiculous. Substrate times time times dilution times, what was the sample type? Sample ID. I don't like that. I'm going to go back up and do some more cleaning here. df names df df1 is sample ID, so I'm going to change that to sample ID with no space in it. Okay. Just to make it easier for us to type, sample ID. All right, that's taken a bit to run. That's a complex model. All right. Summary mod 1, ridiculous. It's testing every possible combination of all of these factors. So our output is absurd. Well, that model's too complex. It defeats the point of a model. It's overfitting to our data. So step AIC is in the, I believe, the mass package. So I'm going to go back up here, library, mass. Step AIC. All right, I'm going to give it our big full model. Okay. How did I pick this model? I just picked every possible explainer and threw it in. Okay. So the question about how to pick something to start this whole step AIC is tricky to answer. You have to think about your data. What in there might actually be explanatory? Okay. In this case, I do think that substrate is explanatory. That's pretty clear. I don't know about dilution. It looks like they're all kind of following the same path. I don't know if they're different enough from each other. So that might be worth taking out, but we'll see if step AIC finds that. Sample ID, I do think that there's a difference between soil and water here. And then time obviously has an influence. So you kind of just have to like plot your data a bit, plot it a few different ways, see what makes sense to you, and use some critical thinking. Okay. If I also had time of day in this data set, what are the odds that that's going to affect what the bacteria are capable of utilizing? Pretty low. And so maybe I would leave that out of my complex model. Okay. So I'm going to save this as step and just let it churn through. And this is going to take a second. It's testing a lot of stuff. Okay. Now let's see what step is. It's a list of 31. We got coefficients, residuals, fitted effects, the R-square values, quartile rank, all these different families, and then there we go, model. So step $, model. No, I don't want that. How do I find the best model?
Is it step $ formula?
There it is. So that's our model. Let's take that, copy, okay, and do mod 2. GLM data equals df formula equals step $ model or formula. There, I don't have to cut and paste. Okay, ANOVA, mod 1, mod 2, residuals, mod 1, squared, the mean of that, okay, so our full model has lower mean squared residual, the error. It's got 0.13, and this reduced model has 0.14. They're really not that different from each other, but this is such a simpler model that it's probably the better one to use. So let's add some predictions. Predictions. And so that will be add predictions. It wants our data and our model. So our data is df. I want to do it with both. So instead of add predictions, I'm going to say gather predictions so I can feed it both models, mod 1 and mod 2. Okay, let's take a look. The add predictions and gather predictions functions take the model, basically all those coefficients and everything, and they tell you what the response would be given a certain set of conditions. So if you remember, let's see, like summary mod 1, it basically says, okay, if you are asparagine and you're in wastewater, given a certain time and a certain dilution, this should be your absorbance based on what the model thinks. Okay, so it uses these coefficients to calculate, kind of like the equation of a best fit line. We're just kind of placing the points onto those lines. All right. So that's what our pred column here is. And what's nice about the add predictions / gather predictions is that it gives you your original data frame plus this new column of predictions. And already I see some pretty stupid predictions on here. Can't have negative absorption, but okay. And then since we use gather predictions, it also gives us another column and tells us which model made that given prediction. Okay. So here in model 1, if you were from Clear Creek with this dilution, that substrate at this time, reality was 0, and it predicted pretty much 0, but a small negative number. Okay.
Let's see. For one thing about predictions, if you wanted to predict, let's say, like a factor, like sample ID. Like, for example, you have all of the other things, but you want to know where a sample came from, kind of thing.
Oh, so in that case, you would have to build your model with substrate or sample ID as the response. So it would be sample ID is a function of all these other things. But
would the prediction be like one of those locations?
No, the prediction, because remember that you're talking about like a logistic regression or an ordinal regression. The prediction would be if you'd said type equals response, the percentage of chance that it thinks it would have to be in Clear Creek.
Okay.
Yeah. It's not going to be able to predict Clear Creek, but it'll give you a percent.
Mhm.
All right, so now let's plot these. So predictions, send it with a pipe to ggplot. Our aesthetic, x equals time, y. Do we want to show reality here? Let's leave the y out for now. Color equals factor dilution. I didn't say factor dilution in my main formula, did I? But it does need to be a factor. So give me a second to correct this mistake. Okay, so they both did a little bit better. Then I'll make my predictions. Okay, color factor dilution plus. All right, so let's show the real data all in black. Geom point. Okay. So we need to give it a Y aesthetic because we didn't give the whole plot a Y aesthetic, so we can just set one inside of geom point. So only it knows about this particular Y, and that's absorbance. And then I'm going to go outside of the aesthetic and say color equals black. And then size equals 0.5, so we'll make them small. Then let's throw on a geom smooth, and we'll say SE equals false, and method equals LM for straight lines, and then we'll give that its own special Y, pred. All right, so does this make sense what we're about to see? Everything is going to have the X aesthetic of time. This geom point only knows about absorbance values and will be black and half size. And then our smoothing function will draw straight lines, and the only Y it knows about is predictions. So our lines are going to show us our model predictions. The black dots in the background will be reality. Oh, I forgot something. Facet wrap as a function of substrate. All right, let's take a look. How'd we do?
I mean, they're just kind of going in the middle of, all the predictions are just in the middle of the black dot, so all right.
Yeah, all right. I agree. It's fine. One thing we did leave out, though, maybe we should make, instead of color dilution, we should say color equals model, because we had two different models in there. So in this way, we can kind of compare both of our models. And they're right on top of each other. Okay. Yeah, they were pretty similar. All right. So this lets us see that our simplified model might technically have a slightly larger mean squared error which it did. It was like 0.003 bigger or something. But that doesn't matter. And so the simple model is still a better one to use. Okay. So now, Cody, when you're saying your predictions didn't look like predictions, is this kind of what you mean?
Yeah, I think so.
Okay. Yeah. It's what's going on here is that this maybe isn't our best model. Another way we could try to do this is change color to substrate, and then sample ID there. Oh boy. So you can see our model is getting some of these higher points here and there, where it didn't look like it when our model predictions were only based on substrate, but it's okay. So each one of these lines represents mathematically how the prediction was calculated. Okay, it found a given row in your data frame and it said, okay, my model knows about these things in the formula. It knows about time, substrate, dilution as a factor, and sample ID. Okay, if we have this time at this substrate in this particular sample, that's what we expect. So it does okay, I suppose. Does that help clear up? Or did I leave something out that you still wanted to know about the ad predictions?
No, I think I just need to mess around with it a little bit more. Like,
okay.
I think this helps.
Your predictions aren't going to be perfect. They never are. Every model is wrong. Some are still useful. That's what we're going for. We're going for useful. Okay, if this helps us make an educated guess about reality, and it does a reasonably good job, it can be useful. It's not going to predict reality all the time because there's other stuff happening in the universe that didn't go into our model because we didn't collect that information. So maybe time of day was important, or maybe different people ran different plates on this biolog plate and they had bubbles in it or something, you know. If we had all that information, we could get better predictions with our model, but we don't. And the fact is, we never will have all the information about any system that we're studying. It's infinite, right? Okay. So as long as they're doing a reasonably good job, that works. Okay. Oh yeah, I promised to talk a bit more about the AOV table. So I threw that out, didn't I? Well, let's make a new one, mod 3. AOV data equals DF. Formula equals absorbance as a function of substrate. Let's call that good. Okay. Summary mod 3. Right. Substrate's important. That's all it can tell us. Don't you want to know which ones are important? Well, we could do a GLM and look at coefficients, or we can do this Tukey test, Tukey HSD, and basically we just feeded our model. And actually, I'm going to save this as Tuk. So let's take a look at Tuk here. Oh dear. What has it done? This only works through categorical data.
Is it giving you a little summary of each substrate? Some kind of information?
Pairs of substrates. Yeah.
Oh, okay.
Every possible pairing, and we got a bunch of them. It might be easier to read if I. This is kind of a lot, so let's change that to sample ID. Just so we, yeah, sample ID is important. Okay. So this might make it easier to see. Soil 1, it finds the mean value of that and subtracts the mean value of Clear Creek. Then soil 2 in Clear Creek, and then wastewater in Clear Creek, and soil 1 and 2, wastewater in soil 1, wastewater in soil 2. So it takes every possible combination of factors, takes their means, and subtracts one from the other. So here it's saying there's a difference in the mean of 0.58. So which one's bigger?
Soil 1.
Great. How about here?
Soil 2 is bigger.
Yeah, soil 2 is bigger. So it's a bit tricky to read. If you see negative values, it means the second one is bigger. But this is nice because it also gives us this plot function. We can just say plot TOOC, and here we go. And it's kind of hard to read them all. So it's not filling them in because they're taking up too much room, but you can see what's happening here. If there is no difference, that's 0. And it's plotting the 95% confidence interval about the two means. So this is similar to what a t-test does, right? It compares the means of two groups and tells us if they're significantly different or not. Okay. So here it's saying soil 2 and soil 1, significantly different or not? Not. The bars cross that 0 line. Okay. Soil 2 minus Clear Creek. Here's the difference of 0.6. Soil 2 is bigger, and we're certain that that's significant because it's not crossing this line. Functionally, this is equivalent to doing a t-test on each one of these pairs individually. However, it does them all at the same time, so you don't run into multiple hypothesis testing. Okay, so these p-values are actually real. All right, and here you see, yep, soil 1 and soil 2. Can't tell if they're different. Okay, here's the amount of difference, the lower 95% confidence, the upper, and then the p-value. Okay. So that's pretty neat. You can't do a Tukey test on a GLM. And so I guess that's a nice little benefit of doing an AOV. If I say time in here also, it gives me a warning. Non-factors ignored, time. So if I look at TOOC, even though time was significant, it's going to give me the same output. So Tukey test will automatically toss out all the non-factors, the non-categorical data. Okay, it's not possible to do this test on them. Okay. Next up on our tour, POSIX CT. So let's see. If I just type in sys_time, that's what it gives me. This is the format of time that R really loves to work with. Okay. It's year hyphen month hyphen 2-digit day, and then 24-hour time in hours, minutes, seconds, and then a time zone. Okay, so that's kind of neat. If I say time 1 is sys time, time 2 is sys time, I can use this little function diff. Diff works with a lot of things. It just tells you the difference. But if you feed it to POSIX CT times, time 1, time 2, it diff time. You can say, okay, here's the time difference. It was 6.9 seconds between typing those two and hitting Control Enter. If I run time 2 again and then diff time, now we're up to 43 seconds. Okay. So being a time class carries some benefits. If you really are talking about time, it's good to set it as time. Let's see here. Library, tidy quant. Okay. And then, what was the function? There's a bunch in here. You can see that I don't know what I'm doing. I've only used this package a couple times. That's it, I'm Googling it. Tidy quant. Get stock index. TQ get, okay. Oops, I forgot to save it. Okay. Does anyone know that stock index?
Forbes?
Yeah, it's Ford. It's just the easiest one to type. Okay. So that gets me the stock index going back 10 years or whatever by default. Okay. So I did this because one of those columns is a date already. Okay, so let's see, plot F. No, it doesn't like that. Okay. Names F. We'll make our own plot. I had to try. ggplot aesthetics. Oh wait, F. Oh, you know what I've done? You can't call anything F. That's a special reserved value. That means false. That's why it turned yellow on me. Oops. Okay, ggplot forward aesthetics x equals date, plot equals close plus geom smooth. All right, so being a date lets you plot things automatically as a date, which is super nice. Okay. Let's see. Let's say you have a column of years. We'll just do 2 of these. Column of months. Oops. And column of days. if I do paste year, month, day, we get our two values, 2020 Jan 13 and 2020 Feb 17. So I'll save that as dates. Okay. Dates as POSIX CT. Womp womp. Character string is not in a standard unambiguous format. So let's take a look at this. Where's the format? It's kind of annoying to use, but it's saved my butt a few times, and I still have to Google it every single time. There it is. Okay. So you can tell it what format your data's in. You don't have to mess around. Okay, so here I can say format is, and then in quotes. Okay, let's see if I can find a nice list. As POSIX CT formats. Have to Google it every single time. Okay. So we've got lowercase y, uppercase y. We've got lowercase m, lowercase d. We've also got like a capital M and then hours and minutes. So let's try that. So in a string, you put a little percent year. And then let's see, we've got a space, right? So we've got full year, the lowercase y is like just 20. Okay. Then a space, and then Jan. So percent capital M, and then a space, percent day. Nope. Time. Here we go. Day of the month as decimal number. Got weeks. Month as decimal number. Abbreviated month, percent B. There, that worked. So it may take a bit of Googling, but if you got 10,000 rows, you just saved yourself a lot of time. So you can go from almost any date format into POSIX CT with this formatting trick here. So basically look up the way you've got it in, if it's Jan, Feb, March, right? Capital B if it's the full month name. Okay, so January would be a capital B. So that gives us our nice POSIX CT formatted, and R knows to treat those as dates now. When you're calculating diff time, I'll just save that as dates over itself. All right, time difference of 35 days. HR. hours? How do I get it to tell me how many hours that is? Once again, Google, let's see here. Units. Oh. Dates 1. Dates 2. I only fed it one date. Okay, 840 hours. So that's kind of neat. Weekdays, dates. It'll look back through the calendar and tell you what day of the week each one of those was. So that can be kind of handy. Any other questions about how to get started working with the time class, or the POSIX CT?
Nope, I think that'll help a lot.
Okay. Yeah, the real trick that kind of got me hooked on it was this. I mean, obviously, I still don't know how to use it off the top of my head. I have to look it up every time, but that's really nice if things are Jan, Feb, March, which they often are. You know, how do you convert that into diff time? It'd be a real pain in the butt, but they give you these little shortcuts. Okay. If you're, let's see, when I pasted those together, Sep equals hyphen, or yeah, hyphen. So now I've got 2020 hyphen Jan hyphen 13. I'd have to tell the format, hey, after that year is a hyphen. Okay. So it's able to do that. So it kind of doesn't matter what formats your dates are in. It doesn't have to be year, month, day either. If it was year, day, month, you could just put these little shortcuts in that order. So you could say Y hyphen day hyphen month.
Yeah.
All right. Now, we got at least 10 minutes, so let me show you. This is where to go for genomic data, NCBI. If you just Google NCBI, it's almost always the first hit. Okay. So the National Center for Biotechnology Information. So let's say you wanted to build a phylogeny of dogs. Well, you can just type in a Latin name of the organism up here. Let's see, what do I want to search? I want to search for nucleotides. I don't care about protein clusters or papers or single nucleotide polymorphisms, any of that. Just give me some nucleotides. Okay. Now we can narrow all this down with these filters. Yep, it's definitely an animal I'm looking for. Okay, what type of molecules? Genomic DNA, RNA? You want mRNA? Yeah, genomic DNA. We're building a phylogeny. Okay. Nucleotide or GSS? Nucleotide. Okay. So here we go. We got a full genome assembly for a boxer named Tasha. That's adorable. Just last month. Neat. Okay. Now here we go. We can customize our search over here in this box. It takes a bit of getting used to, so just play around with these. But like sequence length, we want between 500 and 2,000 bases, let's say. Okay. That's a lot of reads. We got 222,000 different reads in here. Oh dear. Well, what to do? We'd want to go through basically and pick some of these that we wanted to build a phylogeny with. And we'd want it to all be the same gene, otherwise we can't align them. All right, so maybe we should add a gene in here. Let's see. What gene might we want? How about CO1? Maybe that'll get us something. No? Okay. Bingo. 99. Okay. So we've got all these. We can actually just send them to a file. When you do this, you're probably going to want to pick FASTA. Sort by default order, sure, whatever. And presto. We've got the gene sequences for cytochrome oxidase subunit 1 for 99 different dogs. Just like that. Okay. So it makes getting the data pretty straightforward. What to do with that data once you have it is the tricky part. All right, that's sequence.fasta. Wait. I should have downloaded that. FASTA. Save file.
So one question with that is, you have those 99 sequences, right? What if you wanted to do like 2 different genes? How would you kind of get those together by species?
You're going to have to take my bioinformatics class for that one.
Okay.
It's
2021.
yeah, it's tricky command line stuff at that point. However, we can actually read this straight into R as it is, since we just have that one gene. But yes, otherwise we would be concatenating all those sequences together and treating it like it was one big long sequence. Okay.
Yeah. Okay.
Yeah. So let's see. Got my terminal here. And let's move my downloads sequence.fasta to here to play with it, take a look at it. Oh boy. That can't be the same thing. Nope. Okay. Move sequence.fasta. Move downloads sequence. I bet it's that third one. Sequence 3.fasta. Oh, I got to move it here. Okay. Yeah, there we go. Hey, we got some outgroups. No, this is the wrong one too. I downloaded too many files. But we can do FASTA formatter sequence FASTA to clean.fasta. This will put it into pretty form for R. So we got very short read. That's a big package. Short read. There it is. Read FASTA. And we're just going to give it a path. Oops. It's on desktop. Clean.fasta. Okay, so now we've got our reads in here. They're kept in this weird short read format, but that's okay. That's what we want. It takes care of all genomic ranges stuff, all that, which maybe you don't know about, but it's a nice little package, reads it in. So at this point, it's ready to start pumping into some other packages to build a phylogeny. Okay. So if you want to go the phylogenetics route, this is probably your bet. Okay. We're gonna stick you with single gene at this point because you don't have the command line chops to get it all set up otherwise, but that's okay. We're learning. The point of this final project is to learn a new package, right? To kind of build out your R skill set in a direction that you want. Okay. But there are packages for that. The ones I would search for is Fangorn. All right. So the Fangorn package is for building phylogenetic trees. We've got the ape package, analysis of phylogenetic something or other. Which lets us like reconstruct ancestral states, all kinds of stuff. Allows us phylogenetics and evolution. All right, so those are your two main packages, and then the MSA is multiple sequence alignment. And so essentially we just align them, feed that into Fangorn, build a tree, and then we can analyze anything we want about that tree with ape. Okay. And that even lets us use ggplot to plot phylogenetic trees, plot characters onto them. So in and of itself, this information might be interesting to you, but we need to know something else about it, right? So for each one of these, we want metadata, right? Where did the dog come from? Okay, you can download all that too, and then basically extract it with R using some string searching. Let's see here. Boom. So we've got a zip code. So maybe we can look at a phylogeny of dogs in the United States based on zip code and see if there's any phylogenetic signal or biogeographic patterns. Okay. So we can get that kind of stuff out of GenBank, build a data frame, and get a pretty cool analysis. And a month is definitely enough time to do it. And we are out of time now. So I hope I got your questions answered satisfactorily. Is that a word? I don't know. Talking to myself here. All right. Any other questions before we take off? All right. Find that data set. Get your question. Ernie.
So for this Sunday, we have assignment 9 and 10 due? Just 9?
Just 9, yeah.
Okay.
Since we're a bit behind.
Thank you.
But remember 9, you need to have somebody else give you feedback on it.
Do we have to report the feedback they give us or not?
Just put their name on it, like feedback provided by whoever. Yeah.
Cool. And do you want it in the like a separate R file than our file?
No, it's okay. You can just re-edit that one
Okay.
Totally fine. Yeah, just get their feedback, incorporate it, slap their name on it as a little subtitle, and you're good to go. And
Okay.
that's what I'll grade after you've had a chance to kind of tidy things up a bit. Yeah.
Awesome. Thank you.
hmm
Thank you.
Have a good one.